Y'all, today we are covering the principles of vaccinology or vaccines. We're going to talk about what they are, how they work, what's in them, how they're developed. I thought this would be a good thing to cover while we're doing the immune system, and especially given current events with COVID and kind of the race to develop a COVID-19 vaccine or vaccine against the virus SARS-CoV-2 really is what it is. Okay, so let's just get started. So this is an outline of this lecture, so we'll kind of follow along. We're going to talk about what is a vaccine, how do vaccines work, the history of vaccines, what is herd immunity. We hear about herd immunity a lot, especially now in the news. Properties of a good vaccine. We're going to cover types of vaccines, what is in a vaccine, phases of development. We're going to talk about how you get the right immune response. And then I'm just going to very briefly go over myths about vaccines. Okay, so what is a vaccine? Probably everybody knows this, especially because it's in the news, but this is the CDC definition. It is a product that stimulates a person's immune system to produce immunity to a specific disease, protecting the person from that disease. Vaccines are usually administered through needle injections, but can also be administered by mouth or sprayed into the nose. These target the adaptive immune system. So we've been covering the immune system, and you know about the innate immune response and the adaptive immune response. The innate immune system cannot be trained. So what vaccines do is they're targeting the adaptive immune system to train those cells, your B cells and your T cells. And again, these train the immune system to mount a response the first time it encounters a pathogen, like the actual pathogen. So if you get the flu shot and then you're exposed to the influenza virus, your immune system will be able to make an immune response right away. It doesn't need to be infected by the virus to learn. So it kind of mimics that same thing as getting the virus. So I'm sure everybody knows this. I just wanted to mention it. I'm going to mention now too, vaccines can be made against viruses, they can be made against bacteria, and also you can have vaccines against toxins. I am not talking really about how vaccines are made against toxins, but that is something else. Some bacteria produce toxins that cause disease. So the bacteria itself doesn't cause the disease, but the toxins it makes does. And so you can have a vaccine specifically against the toxins that don't affect the bacteria. So that's a whole other thing. I just kind of wanted to keep this simple, but if you're curious about that, let me know and I can find you some good YouTube videos. Okay, so how do vaccines work? Since we've been talking about the immune system, hopefully this is kind of like something you'll just understand. But so you have the vaccine, it's being injected here through the skin, most likely. And then, so here is this, here is the vaccine. It's say this is a dead virus or dead bacteria. A couple of things are going to happen. You are going to have an innate immune response. So that's just always going to happen anytime something gets through our protective barrier. You can't really turn that off, and you don't want to. You're also going to have some B cell activation. So B cells can be activated by native antigen, just by loose antigen in the body. So they're going to be stimulated this way. You're going to have antigen uptake and presentation by those antigen presenting cells, those APCs. So this is a dendritic cell. It's going to go on to like the lymph node, this lymphoid tissue, and it's going to present that antigen to naive T cells and naive CD4 cells and naive CD8 cells. So these two types of T cells. Some of these CD4 cells are going to go on and trigger naive B cells as well. They're going to pass on those cytokines, and you're going to end up with plasma cells that make antibody and memory B cells. And then some of your helper cells are going to go on and they're going to help trigger the stimulation and activation of cytotoxic T cells, your CD8 cells, and you're going to end up with, again, these effector and memory CD8 cells. And you'll end up with memory CD4 cells as well. I should make sure I mention that. And CD4 cells can differentiate into multiple other types of T cells. So we didn't even talk about most of the types of T cells, but there's a lot. So anyway, so this is all of this what's happening when you get a vaccine. So some of this antigen presentation can happen in like the tissue. Some of it's going to happen in your lymphoid organs, like your lymph nodes or your spleen. So we talked about kind of those locations, the lymph nodes and the spleen, where all these cells are. And so that's, where all these cells, where all your B and T cells are. And so this is where this antigen presenting cell is going to do that. In the case of MALT, in your mucosal associated lymphoid tissue, it's going to be happening right there, in that M cell. And then the dendritic cell is going to go on to the lymph node. And we're going to talk about why that's important a little bit later on, because there's kind of differences in your immune system between the mucosal system and kind of everything else. So you're going to hear me in this lecture a lot refer to your mucosal immunity, and that is immunity in MALT tissue. Okay. So just some history of vaccines. I put a lot in here because I always think it's really interesting. I won't really go through all of this in detail, but vaccines have kind of been used for a long time. So in as early as the 1400s, probably earlier, the Chinese were using inoculation to intentionally expose a person to a disease, hoping they would catch a milder form and be spared the serious disease later. So this isn't quite the same thing as vaccination, but it's pretty similar because they're giving the person the disease. So this is one of the ways it was done. This man is actually blowing parts of a smallpox pustule into this child's nose. And I know that sounds real gross, and it pretty much is really gross, but they were much more likely to get a milder form of the disease. Another thing later on, this type of inoculation is called variolation. It was kind of spread around the world, and in Europe they started scratching little bits of smallpox pustules into someone's skin. So they're intentionally giving them smallpox, but then hoping they would not get the really serious form of smallpox later on. And again, that's called variolation. And it comes from the name of the smallpox virus, which is variola. And then late in the 1700s, Edward Jenner, this man, started noticing that milkmaids did not contract smallpox. So this is pretty important. Like, why is this group of people, just milkmaids, not getting smallpox, even when it's raging all around them and people they know have it? Because smallpox was highly infectious. It's extremely, extremely infectious. So in 1796, Edward Jenner decided to test this. So what he did is he inoculated this 8-year-old boy, James Phipps, with cowpox. So he speculated that milkmaids were getting cowpox, which is similar to smallpox. It causes little pustules. It's a disease of cattle. So he hypothesized that because they were getting cowpox, they couldn't get smallpox. So basically, he thought cowpox was inoculating them against smallpox. So he tested it on this poor boy, James Phipps. He was an orphan, so there's kind of definitely some ethical issues there. But then later in the year, what he did is he then challenged Phipps with smallpox. So this is Edward Jenner scratching smallpox into James Phipps's skin to seeing if he would catch the disease, and Phipps remained healthy. He should have gotten smallpox, but because he'd already had the cowpox, he remained healthy. So this is the first use of a vaccine. There was another family that you may read about if you read about this, that the man gave his whole family cowpox and none of them got smallpox, but he never published his work. He never did any more research or documentation, so he's not really given any credit. But Edward Jenner is, and with James Phipps. So I feel like James Phipps should really definitely be remembered. This poor boy who could have gotten smallpox. So here's some just quick more history of vaccines. So around 1802, Thomas Jefferson started advocating them. The first mandatory smallpox vaccination happened in 1853 in the UK. But then here's a lot of information about Louis Pasteur. So Pasteur was really important in kind of the world of vaccines. So he developed the first laboratory vaccine, or vaccine developed in a laboratory, so against chicken cholera. He developed the first successful anthrax vaccine, the first successful rabies vaccine. He was the first person to successfully protect a person against rabies using a vaccine, which is really, really important. Rabies is basically 100% fatal. And it was a big problem back then. We don't think of it as a problem now, mostly because of vaccinations, or actually really entirely because of vaccination. In 1922, we had the first school requirements, so schools requiring vaccination. Again, it was against smallpox. And then the polio vaccine, the first polio vaccine, was developed by Jonas Salk, and it was proven successful in 1955. So some really kind of important history about vaccines. You may not hear a lot about smallpox, but that's because it's been declared eradicated. It was a huge, huge scourge of humans up until 1980. Really until the 70s when the WHO decided that they were going to eradicate smallpox. And the only way they could have done this is because they had a very, very effective vaccine. Smallpox is a disease that's only found in humans, so all the WHO had to do was vaccinate enough humans, and smallpox would eventually be eradicated. And then in 2011, a disease called rinderpest, which was a disease of cattle that especially affected countries in Africa, it was also declared eradicated, again through the use of vaccines. Okay, so on to herd immunity. So we hear all about this in the news, the importance of herd immunity. People are debating with COVID, should we allow there to just be herd immunity naturally? I'm just going to talk about what it is. So herd immunity is when a high percentage of the population is immune to a disease, either through natural exposure or vaccination. And with vaccination, the goal is to protect those that are most vulnerable. So this requires protective immunity. So the vaccine, as we think about developing vaccines, and if we want to get to this point of herd immunity, the vaccine is going to have to be effective. It's going to have to prevent infection by the pathogen. So, and then we also, with herd immunity, we need to know what percent of the population develops a protective immune response from that vaccination. Because, so like in the flu vaccine, it's debated, the effectiveness. It could be as low as like 50%. It could be as high as 75%. If only 50% of the population is getting a protective immune response from that vaccine, then more people need to be immunized to create herd immunity. I hope that makes sense. But in all cases of herd immunity through vaccination, high vaccination rates are a must. So also, if we're thinking about herd immunity developed through natural infection, it would require a high percentage of the population to get the disease. So if it's something deadly, a larger number of people are going to die from if we're trying to get there through natural infection than if we are through vaccination. So this is a little infographic kind of showing herd immunity. So all of the blue people are not immunized but healthy. Yellow people are immunized and healthy. And red people are immunized and they're not immunized, they're sick and they're contagious. So in this scenario, we have a bunch of people that are not immunized, but they're healthy. And we have 2 people here that are not immunized and sick. So if it's a contagious disease, it's going to spread through the population and it's going to reach most of the population. So in this scenario, we have some immunized people, but most are still not immunized, and we have our 2 sick people. So some of the population is again immunized, but still it's not that many. So the disease is going to be able to spread through the population, and it's going to infect, again, pretty much the same amount of people. You might have a few less people infected. So in this scenario, we have most of the population is immunized. There's just a few that aren't. And then we have our two sick people again. So in this scenario, as the disease spreads, it's really not going to be able to spread that much. So here we have a non-immunized person, and they're completely surrounded by immunized people. So basically, the disease couldn't get through that barrier of immunized people to infect them. This person is not immunized, but they're pretty close to a sick person, and that disease was essentially able to get through to them because they're nearby and there weren't as many immunized people kind of in between them and the sick person. So the idea is that if you have a high number of immunized people, they're going to build this like protective barrier around unimmunized people so that the pathogen can't get through to them. Now, if you have direct contact, that's kind of different, but this is in an overall population. So again, the point is that it's the overall population. Some individuals may still get sick, but overall, everyone's protected. So if you think of these people that are unimmunized but still healthy, now imagine they have something like leukemia, and so they can't get vaccinated because they wouldn't be able to mount an immune response, and their immune system is suppressed, like they don't have a good immune system. If they're surrounded by these people who have all been immunized, they're protected. So the goal of that is the goal of herd immunity, is to protect those that are most vulnerable. That includes children, anybody immunocompromised. There are some people that can't get vaccines for various reasons. Someone who, like, say, with HIV. So anyway, so this is herd immunity. If you have questions about it, there's a lot of great videos on YouTube as well that explain it. But it's a really important concept in vaccination because this is how we did things like getting rid of smallpox. And we very nearly eradicated polio. We had very nearly eradicated measles. It's on the rise again because there's less vaccination than there used to be. So anyway, that's all herd immunity. Okay, I'm trying to keep this like quick, but I'm sure I'm going to go long. All right, so talking about measles and polio, I just wanted to show some data about how effective vaccines are and how effective herd immunity is. So here we have measles, and this is from 1950 to 2010. And here we have a little 97 to 2011. Because this is overall, this is a very big scale. I mean, this is, what is that? I can't see. 1 million people, or 100,000 people, sorry, 100,000. So in the 1950s, up here in like 1956, we had a lot of cases. It's almost 800,000 cases. And they had kind of started to drop. And one of the reasons they started to drop was isolation. So during this time, this also happened with polio, some things like public pools were closed. Anytime you'd have gatherings of children, because measles and polio really affect children, a lot of those were closed down. So cases naturally started to decline because they weren't able to like pass from child to child when you'd have like a gathering. Public pools was like a huge way that polio was spread. But here at this point, you have the vaccine licensed. And you can just see, I mean, it was already on the decline, but it was not declining that much. It was probably going to hover around 400,000. And then we get a vaccine and it just plummets. At this time, this is about like 1990-ish, is when they started recommending a second dose of the measles vaccine. So this would boost your immunity again. So you'd have 2 doses, you would have a real strong immune response against the virus, and it really just, there's no more blips. It just about eradicated it. So we went from, what year this is, 1958-ish, nearly 800,000 cases, to 1997 when we had less than 150. Now, vaccine rates have started to decline, and so since about like 2007 or so, we have actually seen cases on the rise. And I couldn't find a good graph for the past 10 years, but they have definitely kind of gone up again, and worldwide measles cases are on the rise. So with polio, um, we can see that, and polio has been eradicated in the United States. So even though vaccine rates are declining in the United States, polio has not kind of made a reemergence. But you can see here the spikes of polio, which are really going up here in like 1953, almost 60,000 cases. Polio was terrible, it was very debilitating. It could paralyze people. And so you may have seen about like an iron lung. People had to live in breathing machines because they were paralyzed for very long periods of time. We even had a president, Franklin Delano Roosevelt, who was paralyzed by polio. So here in 1955, when the polio vaccine was introduced, there were just under 30,000 cases that year. And you can just see, I don't know what happened with this blip, but you can see it just plummets. And so now we are down to essentially, well, it's been eradicated in the United States. So 0 cases a year in the United States. Okay, so now we've covered how vaccines, what they are, how they work, the history, and herd immunity. So I'm going to start talking about properties of vaccines, and we're going to go through the types of vaccines, and then what all is in a vaccine. So all the other things. So we know a vaccine is like going to have some sort of pathogen, like a virus or bacteria, but what is all the other stuff? So we're going to go through that now. Okay, the properties of a good vaccine. It's pretty simple. So it must elicit the appropriate immune response. So that is the type of immune response that will work against the particular pathogen. So a vaccine that stimulates the wrong immune response, it may stimulate a real strong immune response, like your body may start pumping out a bunch of antibodies, but if they don't work against the pathogen, it's no good. So just because the immune system has a response doesn't mean it's the appropriate or right response. And that's kind of a key thing to remember when we talk about vaccines. That's why it's hard to develop a vaccine, is because you have to get that immune response just right. A good vaccine would also provide long-term protection. So you don't want a vaccine that only lasts 6 months, because you'd have to be getting a shot twice a year, right? So you want something that lasts a long time, preferably like the length of your life. If a vaccine can protect you for your entire life, that's awesome. It should be safe, obviously. That should, goes without saying, hopefully. And it should be stable. So stable means it lasts over time, and particularly that it doesn't require a cold chain. So a cold chain, I put some terminology at the end of this, but a cold chain is a supply chain that maintains a cold temperature. So a vaccine may require, if a vaccine requires a cold chain, and say someone in the Democratic Republic of Congo needs that vaccine, the vaccine is going to get shipped from the United States. It's going to have to be refrigerated or frozen the entire time. When it gets to Africa, when it gets to the DRC, it's going to have to be refrigerated or frozen the entire time, the whole way to wherever that person lives. Now, the DRC has been in a basically constant civil war for a long time. So it's hard to maintain something like a cold chain in a country at war. So a good vaccine wouldn't require that, because that's hard to do. I mean, think of like putting coolers on a truck, and you've got to have dry ice, or you have to have an electric cooler. So good vaccines are stable at room temperature. They can even if they get warm, it's not a big deal. They should also be inexpensive. This goes without saying. Think about COVID. We need to develop, millions and millions of vaccine doses really, really quickly. If they were expensive, that would be harder to do. Okay, so now we're going to go through the types of vaccines. So we're going to talk about live attenuated vaccines, inactivated vaccines, recombinant vaccines, DNA vaccines, and polysaccharide vaccines. And this is out of order. We're going to talk about DNA last. Okay, so live attenuated. And I apologize for these wordy slides. I just tried to put as much information for you on there as I could. So a live attenuated vaccine is derived from a wild virus or bacteria. So it's taken from the actual pathogen that's infecting people, out in the wild, everyday life. And then it's been attenuated, which means weakened. So this usually happens by them passaging the pathogen through a lot of different cell cultures, and it weakens it over time. But there's other ways. They can irradiate it. They can genetically modify it to be weak. But in any case, it's weakened, and that means it does not cause the disease anymore. So it's not gonna, if it's gonna replicate in your body, but it's not gonna cause the disease. So it's not gonna cause all the problems. So, and for a live attenuated vaccine to create a protective immune response, it must replicate in the vaccinated person. So if you get the chickenpox vaccine, for example, varicella, it's a live attenuated virus. So it's a weakened virus. It's not going to cause chickenpox, but it is going to replicate in your body, and that kind of gives your immune system something to attack. They usually do not cause disease, but very, very rarely a vaccinated person may experience a mild form of the disease. That is extremely rarely, and it's typically in immunocompromised patients. And in an immunocompromised patient, that even though it's not really a pathogenic virus or bacteria anymore, it can replicate unchecked, and it can cause a severe infection just because it's replicating a lot, and it's going to cause a lot of inflammation, a lot of activation of your innate immune system, and that alone can cause a lot of problems. But that is, again, in immunocompromised patients, people with HIV and leukemia, or, for example, it could be other reasons. These vaccines can theoretically revert to wild type. So this is in theory. So this hasn't happened except for one case. Well, it's not even really, but there's a live polio virus, and in some areas of the world, there is circulating polio virus from the vaccine. It doesn't cause the same type of polio as you kind of think of, people being paralyzed or living in iron lungs. So it's not really true wild type polio, but it can spread. So that is kind of a downside of these, is that you can pass on that virus or bacteria, that live virus or bacteria that you've been infected with for a short period of time. So it's not for like ever. If you've gotten a live vaccine, you're not going to pass it on forever. But your immune system is eventually going to come in, going to attack it. You're going to get over the infection. And so then you're not infectious anymore. It's just like a natural infection. So going through this again, speaking of natural infections, so live attenuated vaccines cause a virtually identical immune response to that produced by a natural infection. And that's a really good thing, because we know then that it's going to be the right type of immune response. There are some issues with them. Circulating antibody from sources like transplacental transfer, so the IgG that a baby, a fetus, gets from its mother through the placenta, if it's still circulating in that baby's body, that could interfere with the development of immune response, and so that's known as vaccine failure. This is why we don't give infants live attenuated vaccines. And these vaccines are pretty fragile. They're going to require a cold chain, or they have to be stored properly, like in the dark. So examples of live attenuated vaccines would be MMR, varicella, there's live rotavirus vaccines, typhoid vaccines, and then there's a live intranasal influenza vaccine, an inhaled influenza vaccine. Okay, so just to go over that again, but kind of pointing out the pros and cons, because there's going to be pros and cons to all these types of vaccines. So pros, these are excellent stimulation of the immune system. So they really do stimulate your immune system. They get that right type of that appropriate immune response. They can stimulate cellular immunity, which is really important. That would be immunity from your T cells, especially your cytotoxic T cells. Which are important in intracellular infection. So anything like a virus or a bacteria that's going into your cells, you really need a cytotoxic T cell response. These also stimulate lifelong immunity with only 1 or 2 doses, and they can be given through natural routes of infection. So this is important because this can create immunity in the mucosal areas, so in your malt tissues. And you really need that because injected vaccines are not going to stimulate your malt as well. And I'm going to mention that again later on. Um, and so this is kind of a pro, kind of a con. I mentioned that these live viruses and bacteria can spread from animal to animal or person to person, and that can pass on immunity through a herd. So it's kind of like in livestock, it's kind of used as a good thing. In people, it could be a bad thing if you were, say, around someone who's immunocompromised, yeah, basically someone who's immunocompromised. I don't know where I was going in my head there. Anyway, so sometimes with these vaccines, they will recommend don't be around someone who has been who is immunocompromised. So not that we really talk about smallpox a lot anymore, but the smallpox vaccine is an example of that. Because basically no one in the world anymore has good immunity to smallpox. Um, if someone was to get vaccinated with a smallpox vaccine, they would need to be careful being around other people because they could pass it on. Now, it's not the disease-causing form of smallpox, but they could still pass it on. And you don't want to do that because if it reverted to wild type, that'd be really bad. But this is a totally theoretical example because we don't have smallpox in the world anymore. Okay, so anyway, I get really into vaccines and I nerd out on this stuff, so if I go off on crazy tangents, I apologize. Anyway, cons of these live attenuated vaccines. That theoretical potential to revert to wild type. It's in theory, but it's still something to be aware of. And they can rarely cause mild disease, again, really in immunocompromised people. Most of them really just can't be given to immunocompromised persons. They really can't be given to infants because you could have vaccine failure if the infant has circulating antibodies from their mother. Again, yeah, so that's there. So they're pretty fragile, so that cold chain is definitely a con. And they're not as easily produced, so they're more expensive because you have to grow them. So growing influenza requires eggs. You can imagine like growing live Ebola virus has to be in a biohazard level 4 lab. That's expensive. So it's not as easy to make these. Even though that great immune response, they're not easy to make. Okay, so inactivated vaccines. These are or killed vaccines. Sometimes you'll see them killed virus or killed bacteria or inactivated. These are made from killed or deactivated pathogen. And that can be whole or broken into specific antigens. So sometimes you'll see those referred to as fractional vaccines or subunit vaccines. When it's just parts of a bacteria or parts of a virus. These can be freeze-dried. They're not very fragile, so there's no cold chain, so that makes them very stable. The pathogens cannot replicate. Is it because you're injecting something that's completely deactivated, it's dead, or it's broken into pieces. And the antigens are less susceptible to circulating antibodies. So if an infant can get this because those antigens are not as susceptible to the circulating antibody that they have from their mother. It does require multiple doses to stimulate sufficient immunity. So that's probably going to be 2, 3, 4. I think my rabies vaccination, I think I had 5. I can't remember. The immune response is mostly going to be via antibody. There's going to be very little cellular immunity, especially very little cytotoxic T cell response. So this is important when we remember, some things are intracellular, and you definitely really want that cellular immunity. So there's ways that they can be combined with other antigens to stimulate cellular immunity, but in general, they're mostly going to stimulate humoral immunity, antibody immunity. And antibodies tend to diminish over time, so these are not long-lasting vaccines. And And they're available as, again, as whole inactivated pathogen or fractional or subunit pathogen or subunit. So the pathogen being broken down into specific antigens. And they're most often administered with an adjuvant, so something that's added to stimulate the immune system. They will not stimulate the immune system enough on their own. So some examples of whole inactivated pathogen would be polio, hepatitis A, and rabies. Some fractional vaccines would be influenza, anthrax, Japanese encephalitis. Interestingly, I have had all of these on the screen except for I've never been vaccinated for anthrax. And then the anthrax vaccine is not long lasting anyway. Okay, so pros and cons. Pros, they can be freeze dried, no cold chain. That goes back to that, principles or what's the word? I'm forgot the word. What's the word? Properties. Sorry. It's been a long day. Properties of a good vaccine is that being stable. They cannot cause disease. So they can't even cause a mild form of the disease because they're inactivated. They can be given to infants and those that are immunocompromised. They can stimulate a strong antibody response. So if it's an antibody response that you're going for, if that's the appropriate immune response for that pathogen, that's great. And then you may only need part of the pathogen, and this actually makes them less expensive and kind of easier to produce than the live attenuated vaccines. So some cons, you're going to need multiple doses. They don't trigger cellular immunity, especially that cytotoxic T cell and memory from the cytotoxic T cells. The antibody titers are going to diminish over time. You're going to require boosters over time. You're going to have to get revaccinated as you get older. They're not given through natural infection routes. These are typically injected. So they may not stimulate mucosal immunity. So if it's a pathogen that attacks mucosal tissue, this isn't so great. And it can be difficult to identify the specific antigen that triggers immunity, especially for fractional and subunit vaccines. So it takes a lot more time to kind of develop one of these vaccines than it does a live attenuated vaccine, especially if it's going to be a fractional or subunit vaccine. And again, this kind of is the same thing as in live vaccines, but cultivation of the pathogen for inactivation may be difficult. So this is, I guess, is kind of a pro and a con. So if it's something difficult to cultivate, like Ebola, and you're trying to use a whole killed virus, that's going to be kind of difficult and expensive. All right, so polysaccharide vaccines. These are made of long chains of sugar molecules from bacterial capsules. So these are vaccines for bacteria only. And these stimulate B cells independent of T cells. So if you remember from the immunology lecture, B cells really need those helper T cells to get them activated and for them to class switch. So for them to start producing IgG type antibodies rather than IgM. So this polysaccharide vaccines, they don't stimulate T cells. So they're going to stimulate your B cells, but there's not going to be helper T cells to come along and send the cytokines across to those B cells to tell them to make IgG. So these are not going to elicit as strong of an immune response. And they may not be consistently immunogenic. So immunogenic means the ability to create an immune response. So these might not be consistently immunogenic. It depends on the person. And again, the antibodies against these polysaccharides are less functional than those against proteins. And that's because they're producing mostly that IgM. If you're curious about all of this, I want to talk about with IgM and IgG and class switching. Let me know. It's really pretty interesting. But for now, you don't have to know all of those details. So these polysaccharides may need to be conjugated or combined with a protein to stimulate that T cell response. So you can get a T cell response they're kind of made right. You have to conjugate them with a protein, but they're still kind of going to be a weaker vaccine. And a curious thing about these is multiple doses does not increase the immune response. So if you have 5 doses versus 1, you're going to have the same immune response. So some examples of these, they're not used as often, but Salmonella typhi type 6, pneumococcal diseases, and meningococcal diseases have some polysaccharide vaccines. All right, so again, just going over the pros and cons. Pros, they can stimulate B cells independent of T cells. So if you need an antibody response, that's really good. That's also a con. So I should have put that over here too. They're stable. They don't require a cold chain, so that's always good. They're inexpensive. That's great. They're easy to produce. There's no infectious agents. So they can't trigger disease. And they can be given to infants and immunocompromised persons. So cons, they only work for bacteria. They don't trigger T cells unless they're conjugated with a protein. Basically what you get is what you get. So whatever you get, immune response you get on that first dose of vaccine is the immune response you're going to get no matter how many other doses you receive. And they're mostly triggering an IgM response, which is a weak antibody. So IgG is the good, powerful antibody, and these are mostly going to have IgM. All right, recombinant vaccines. So this is kind of an interesting technology, and I'm going to go through it. I just want to say these are antigens for these vaccines are produced through genetic engineering. And I know that sounds scary. We hear about GMOs in the news, and it sounds like something weird and like from a sci-fi movie, but it's really not. And we're going to go through it. They're actually very safe vaccines. So genetic engineering in this case, and in a lot of cases, is very safe. Okay, so the pathogen itself may be engineered to replicate abnormally. So there's a couple different, so I'm going to go through these types of recombinant. So this one, the pathogen itself is engineered. So it has had different genes put into it or taken out of it. So the example would be there's a live influenza vaccine that is genetically modified to only replicate in the nasopharynx, but not the lungs. So this influenza vaccine, you inhale it, and it's a live vaccine, and it's going to replicate in your nasopharynx. It's not going to cause disease, and it's not going to move on to your lungs where the worst disease response to influenza is. It's just going to replicate in your nasopharynx, and it's going to stimulate an immune response in that malt, in your mucosal-associated lymphoid tissue in your nose, which is also going to transfer immune response, that immunity, to all other mucosal areas, all other malt. So that's great. That's a kind of great trait of that vaccine. Another way these recombinant vaccines are made is the gene encoding for the major antigen. So whatever antigen causes an immune response. So it could be like a major surface protein. It is cloned or inserted into the genome of a non-virulent vector. So examples, the most common things would be yeast or a non-virulent virus. So there are some viruses that we can get that don't cause any disease in us. And so we can actually then use those in recombinant vaccines. And I'm going to go through how each of those are done. Because it sounds complicated, but it's not. It's actually not that complicated. So these are kind of interesting vaccines because the potential from genes from multiple pathogens could be used in the same vector. So one vector, one non-virulent virus, could carry essentially the genes to make antigens for multiple pathogens. So one vaccine could, without being mixed, could stimulate immune response to multiple pathogens. So that would reduce cost and storage and everything, and the number of times you have to get shot if you don't like those. I have a bunch of tattoos, so shots don't bother me. Anyway, So these vaccines can trigger both humoral and cellular immune response, which is great. They can trigger that, again, those B cells as humoral with antibodies, or cellular, which would be your CD4 and your CD8 T cells. They can induce mucosal immunity because you can have inhaled recombinant vaccines. They're being given in the natural route of infection. But live vaccines, again, can always cause disease in immunocompromised persons. Now, it's not going to give an immunocompromised person influenza, but if it's replicating, it can cause an immune response and you can have some mild disease. Examples of recombinant vaccines. This is supposed to be Hep B. Let me fix this. Okay, hepatitis B, HPV, that's human papillomavirus, influenza. Man, I really can't type, can I? Live typhoid vaccine is recombinant, and then the live rotavirus vaccine is recombinant. Okay, so I'm going to go through how this is one that's would be a viral vector. So the first thing I want to point out are plasmids. We're going to talk about plasmids. A plasmid is a round piece of DNA that is that in some viruses, in some bacteria, it's not the main DNA that codes for general life of that bacteria. These are just like extra pieces of DNA in bacteria. And we've actually learned how to use these and kind of harness them in the lab, because plasmids can get passed from bacteria to bacteria, from cell to cell. So this is DNA that can be passed on easily from one living bacteria to another without replication. Okay, so first step in a recombinant vaccine where we're going to end up with a viral vector is the antigen. The antigen of interest has to be identified. That's always going to be the first step in any vaccine. And then they have to isolate the gene coding for that antigen. So they have to find the specific gene. So then what they do is they take a plasmid and they actually insert that gene into the plasmid. Don't have to worry about all the laboratory steps on how to do that. It's pretty interesting though. I've actually gotten to do this in the lab. So working with plasmids, and it's called cloning. You're putting the gene inserting it into the plasmid, and they call it cloning. It's different than cloning like Dolly the sheep. Okay, then the viral vector and the plasmid are introduced to a host cell. So this would be a cell in cell culture. So this isn't like in a living human. This is just cells growing in a petri dish. While they're in there, the plasmid is going to combine with the viral DNA, and then it's going to replicate this virus. So this is a vaccinia virus. So this is, in this example, this image, it's a chickenpox virus. So when that virus is then kind of released from the cell after it's been replicated, it's going to have that gene. So now you have a vaccinia virus that expresses that foreign antigen. So in this case, it's a vaccinia virus. It could be an adenovirus, a cold virus. It could be a number of things. And a lot of times these viruses are either naturally not pathogenic or they have been modified so they aren't pathogenic. And they're going to carry this protein. So then if you get this vaccine, if this vaccine is injected into your arm, this virus is going to start replicating. As it's replicating, this gene on the virus is going to tell your own cells to make the antigen. So they're going to start producing this little triangle antigen, and that is what causes the immune response. I hope that made sense and was simple. Try to simplify it. All right. So this is how a recombinant vaccine would be made if we were using yeast. So they're not injecting yeast into the body, but they're using yeast to make the protein for us. So what happens here is, again, that antigen of interest is identified. The gene coding for that antigen is isolated. The plasmid is combined with that antigen gene. So you have here recombinant DNA. They call it recombinant. It's just combining it. And then that plasmid is inserted into a yeast cell. And then that yeast cell is just fermented. So just like beer, so that yeast is grown and it replicates. And as it replicates, it's going to have that recombinant, that plasmid with that gene to produce that antigen. So then what they do is they filter this all out. They're going to extract that specific antigen, and they're going to make the vaccine. So rather than having the hepatitis B virus in cell culture making that antigen, they just use yeast, and the yeast is just pumping out that protein. And then it can be purified to make the vaccine, and it would be basically a subunit vaccine or a fractional inactivated vaccine. So kind of all of these vaccine types, oops, and the one I did, they kind of work together. So this is technically a recombinant vaccine because it's made with recombinant DNA, but it is also an inactivated or killed type vaccine. Hopefully that makes sense. Okay, so pros and cons of recombinant vaccines. The pros, there's going to be similar stimulation of the immune system as a natural pathogen, especially in live viruses like that influenza one. It can trigger cellular immunity, and very importantly, it can trigger mucosal immunity. It allows for the direction of the immune response to be kind of targeted. So if we know that an antigen, a very specific antigen, makes a very specific immune response, like a TC cell response, a cytotoxic T cell response, we can use that antigen to kind of pump up that immune response. This becomes important because it is so difficult sometimes to find just the right antigen to make the right immune response to attack a kind of living wild pathogen. The vectors have intrinsic adjuvant properties. So adjuvants are something that stimulate an immune response. And if you're using a viral vector recombinant vaccine, it is already going to stimulate immune response just because it's a virus that's entering the body. So if it's also making that antigen, it's kind of like it's going to stimulate an extra immune response just like that. So you're not going to have to add any more adjuvants. We're going to talk about adjuvants when we talk about additives to vaccines. These are going to trigger a long-lasting immune response, so that's always important. They're fast to produce and they're easy to modify. Especially like this, you can make a ton of that antigen really, really quickly just because it's in yeast. And it's very, very easy to modify it just by inserting a different gene into the plasmid and kind of into the yeast and starting over again. And these are very safe. So some of these don't have an infective agent like in live attenuated vaccines. Cons, they're limited to protein antigens. So typically proteins are going to be the most immunogenic anyway, but if you have a bacteria that you need an immune response to a specific polysaccharide, you can't use this method. These are unfortunately less efficient in large animals and humans. So recombinant vaccines work really, really well in mice in the laboratory, but they don't work as well when they get to us, unfortunately. You do need multiple doses. Even though it's a long-lasting immune response, you will need multiple doses to make sure you get a strong immune response. You may need adjuvants if it's not with a viral vector. And live recombinant vaccines are fragile and may require a cold chain. All right, so we're going to talk about DNA vaccines. So this is very, very new technology. This is actually not yet licensed for human use. I mentioned that down on the bottom. So DNA vaccines are plasmids containing DNA encoding for a specific antigen, and they're directly so those plasmids are directly injected as the vaccine. So if we go back to, I keep hitting the wrong button. Sorry, y'all. I am spaz today. Okay, so these are the plasmids. And remember Here, they're recombinant DNA. Instead of inserting them into a viral vector or using yeast to make that antigen, this itself, the plasmid, that recombinant plasmid itself, is injected as the vaccine. They rely on in situ production of antigen, so on site. So once that plasmid is injected, like say into your arm, it's the antigen, your body cells are going to take up those plasmids, and it's going to require that your body makes those antigens right there. If for some reason it doesn't work, you're going to have vaccine failure. These can stimulate both cellular and humoral immunity. They induce long-term immune responses. They're very stable. They don't require a cold chain. There's no infectious agent, so there's nothing to cause disease. They do require adjuvants, and they're limited to protein antigens. Plasmids can only encode for making proteins. There's some problems. There's the possibility of tolerance to the antigen. So there's the possibility, because your own cells start making this antigen, that your body will instead learn that that antigen is okay, which is you don't want. It's the exact opposite of what a vaccine should do. And there's the possibility of inducing antibodies against DNA. So this you don't want. Your antibodies wouldn't be going into your cells and attacking your DNA in the nucleus, but sometimes there's extracellular DNA, and you don't want your body necessarily to start attacking that. Your body already doesn't like extracellular DNA, but you don't want to make antibodies against DNA. And again, this is a very, very new technology. So all of this is still being studied. They're still perfecting it. And it's not, none of these have been yet licensed for human use. Okay, pros and cons. They're actually really inexpensive. Plasmids are easy, recombinant plasmids are easy to make. I did it in lab as a student, and it worked. No, I wasn't making a DNA vaccine, but I was able to make a plasmid that we then put into E. coli, and it made the protein we wanted. They're very stable. There's no infectious agent, so they're very safe. They may be given to infants and immunocompromised persons. There's some cellular and humoral immune response are both elicited, which is good, and long-lasting immunity. Now again, these are not licensed for human use, so a lot of this is kind of in theory. Cons, again, no human use yet. And so far, they've only been tested in veterinary applications, but it does really look like this is going to be something that could be helpful in the future. But they may trigger that antibody response against DNA. They may induce tolerance to the antigens. Oh, and it requires adjuvants. I'm glad I am going through this PowerPoint before I posted it, because I clearly made a lot of typos. Okay, so now we've been through quite a lot of this. I know this is a long lecture still. So we're going to talk about what is in a vaccine, the phases of development, how to get the right immune response, or just kind of some concepts by getting the right immune response, and then myths. Okay, so what is in a vaccine? So this is the kind of list. Besides the killed pathogen or the live attenuated pathogen or the recombinant protein, this is what's in a vaccine. We have preservatives, adjuvants, stabilizers, residual cell culture material, residual inactivating ingredients, and residual antibiotics. All right, so preservatives. These are really important. They prevent bacterial and fungal contamination. So, for example, in 1916, during a test of a typhoid vaccine, 4 children died from bacterial infections because the vaccine was contaminated. So these have been required in multi-dose vials of vaccines since the 1930s. So single-dose vials of vaccines don't have to have a preservative, but multi-dose vials do. This is because you're going in and out of that vial with a needle, and you can introduce contamination. So this picture, you'll see here, it says sterile water. I thought somewhere it says contains no preservative, but that's because it's a single dose. There are 3 preservatives that are used in vaccines in the United States. Phenol, 2-phenoxethanol, 2-phenoxethanol, sorry, I can look at these and just not pronounce them. And then thimerosal. And you've probably heard of thimerosal because it's the most controversial one. So it was removed from childhood vaccines in 2001 simply as a precaution. So Congress was going through and looking at things that contained mercury because there was really a concern about people ingesting too much mercury. So thimerosal contains ethyl mercury, so it was removed again as a precaution. That doesn't mean it's unsafe. So thimerosal breaks down into ethyl mercury and thiosalicylate. I can't say these words. I apologize. Anyway, so methyl mercury, if you've heard of mercury poisoning, it's from methyl mercury. It is known to be unsafe, and this is the methyl mercury is what causes neurological issues and all of the diseases associated with mercury. Ethyl mercury, there is a lot less known about it, but what we do know is that it only causes damage at doses, that's actually supposed to say 1,000. See all these typos. 1,000 to 1 million times higher than in vaccines. And ethyl mercury and methyl mercury break down differently. So you actually, methyl mercury is what you can get stored in your tissues so it can build up over time. Ethyl mercury, your body actually gets rid of pretty quickly, like about within a week or two. So thimerosal was removed, again, simply as a precaution. And what Congress thought was if they removed this thimerosal, it would make vaccines seem safer to people. Unfortunately, it actually seems to have had the opposite effect. And now people are worried about vaccines because they removed this. But again, it was just removed as a precaution. I know I'm stressing this a lot, but it's just because it was controversial, but a lot of people don't understand what thimerosal is, why it was removed. Okay, so what is in vaccines? We're going on to adjuvants. So adjuvants are just something that boosts the immune response to a vaccine. Reactions to vaccines with adjuvants can be stronger than those without, which means you can actually have you can have some health effects from it. You could feel sick. But while it kind of sucks if you get a fever or chills after you get a vaccine, that's there because the adjuvant is making sure your body has a good immune response. So it sucks for a couple days, but in the long term, it's a good thing. You can have local reactions to adjuvants, which would just be irritation, inflammation. So if you've ever gotten a vaccine and it's swollen or gotten red, that's from your innate immune response kicking in. And then systemic reactions would be fever, chills, body aches. So if you've ever gotten a vaccine and had these happen, you're not getting the disease that you were vaccinated against. It's just the adjuvants kind of doing what they're supposed to do and causing an immune response. So in the US, these are the licensed adjuvants. The most important ones to remember are aluminum salts. They're used in almost everything that has an adjuvant. We have a lot of vaccines that don't have adjuvants, so adjuvants aren't used in everything. So aluminum salts aren't used in all vaccines, but they are used in the DTaP and Tdap vaccines, hepatitis A, HPV, meningococcus B, Hib vaccines, and it scares people because it's aluminum, and they think they're injecting this metal into their body and it's going to make them sick. But these have been used for over 70 years, and they're actually very, very safe. It just causes an immune response, which is what you want because you want your body to be able to fight off the disease. So these are some other adjuvants. I just put them in because I thought some people might want to know, but they're much less common. So AS04 or AS04, MF59, AS01. This one's actually interesting because it's made from this monophospholipid A, which is from bacteria, and it's combined with this natural compound from a Chilean soapbark tree. It's only used in the Shingrix, the shingles vaccine in the United States, but it's something that could be used more in the future. And then this CpG 1018 is a synthetic form of DNA. It's only used in one of the Hep B vaccines. So aluminum salts are used the most common. There's 4 different types of aluminum salts that may be used. But you may in the future see a vaccine with one of these adjuvants. And it all comes back to the type of immune response you're trying to elicit with a given vaccine. So these can help, these other types of adjuvants can help the body make that specific immune response. All right, what else is in vaccines? We have stabilizers. These are really important to keep the vaccine stable after manufacture. You don't want it to go through any changes. You don't want the proteins to change. And so those active ingredients, because proteins can denature, they can unfold if they get too hot. So these are just something that keeps that stable. And so it reduces the need for a cold chain, which is very, very important. Cold chains are easy to maintain in like the United States, but they're very hard to maintain in other parts of the world. So we like these stabilizers. And they're pretty simple things anyway. The most common ones are just sugar and gelatin. Now, gelatin is an animal protein, so in rare cases, it can cause an allergic reaction. All right, so residual cell culture material. So this is the material used when growing a vaccine. And it's really just difficult to remove 100% of it. But it's all pretty much non-toxic. It's like sterile saline or eggs, actually, egg proteins. So really, it's generally harmless. But unless you have an egg allergy. So eggs are used to grow vaccines for influenza and yellow fever. So influenza virus is very hard to grow, but it grows well in eggs. So this is what they do. They actually inject a little bit of influenza into an egg, and then they let it grow, and then they go back later and they extract it. I've actually gotten to do this too. It was really fun. It wasn't for a vaccine, it was just for influenza research. But there may be some leftover egg proteins, and in some people that can cause an allergic reaction. This isn't all influenza vaccines, but this is why it's kind of important to get a vaccine in a medical setting and let the doctor know if you are allergic to eggs. Okay, so residual inactivating ingredients. So these are ingredients that are used to inactivate those pathogens for inactivated vaccines. So if it's a virus or a bacteria, they're in very, very small quantities. And these inactivating ingredients are typically diluted. I mean, they're filtered out and they're diluted, but you may still have a tiny amount left. So the one that always scares people is formaldehyde. But it sounds scary, but it's actually a really common substance we encounter like every day. So if you've ever bought a new mattress or new furniture, there's formaldehyde in it. We have just naturally occurring amounts of formaldehyde in our body, and much higher amounts than would be in a vaccine. So one of the kind of best examples is pears. So pears, a 200-gram pear contains up to 12,000 micrograms of formaldehyde just naturally. And this would be in an organic pear too. I mean, it's just there. Vaccines contain up to 100 micrograms of formaldehyde. So that is 0.83% of the amount that would be in one pear. So if you eat one pear a year, you're getting more formaldehyde than you would from all the vaccines you get that year combined. So it sounds scary until you kind of learn about it, and then it's like, oh, it's everywhere. Then there can be some residual antibiotics in vaccines. These are just used to prevent contamination during manufacturing. So these are really important because any contamination in vaccine manufacturing can be deadly. Remember, we saw the 4 children that died in 1916, and there's many other cases of this happening, I should say. Vaccine contamination is a problem. That's why there's preservatives, and that's why it's grown with antibiotics. Again, it's very, very small quantities. They make vaccines safer. So some people are scared of it, but it makes vaccines safer for everyone. And the antibiotics they use are not going to be the ones that people are commonly allergic to, like penicillin. So they're not going to use penicillin because they know a lot of people are allergic to it. So a really common one is neomycin. This is the one we used in lab when I was in school. Okay, so we've gone through all of this. We're on to phases of development. Thank you all for sticking with me. I love this stuff, so I'm really excited to teach it this semester. So these are all the basic stages of vaccine development. There's the exploratory phase, preclinical, clinical development, regulatory review and approval, manufacturing, and then quality control. Really, because this is A&P and we're covering the immune system, I'm only going to talk about the exploratory and preclinical phases of vaccine development. Oh, I got to take a sip of water. If you are interested in these other phases, you can Google. The CDC website has a lot of information. I stole this infographic from the Masters of Public Health program at George Washington University. So you can kind of look them up, or you can just ask me and I will be happy to give you more information. Okay, so the exploratory phase. So this is when researchers are identifying the antigens. So they could be natural or synthetic. They could be making antigens and seeing if they're similar to, this is actually Ebola virus, seeing if they're similar to the natural antigens and if they would work. So during this phase, they will also isolate the antigen producing genes. So what gene of the genome of that pathogen produces that specific antigen, that protein. And this is basic laboratory research. So this is just really basic experiments identifying these antigens. These are typically government and grant funded. This often happens at universities. You have a lot of students actually doing this work. But this phase of development can last years. It can take 4 years or more to discover all of the antigens that involves the pathogen. And that's not even discovering if they work to elicit an immune response. This is just figuring out what they are. Nowadays, we have a lot of technology. We can do this a lot faster. We saw this with COVID. It took much, much less time to discover everything about SARS-CoV-2 than it did SARS back in the early 2000s. Ebola was first discovered in 1976, and it really took until 2016 until we even had a really good understanding of all of the antigens on Ebola, and it's a pretty simple virus, actually. So when we talk about antigens, so these would be the antigens. All of these proteins could be antigens. All of these little pieces of a protein could be an antigen. So the major protein on Ebola is this glycoprotein. It's called GP40. And so the identification of GP40, what is it? And then it's also finding out all the little pieces of GP40 that would be the epitopes that could cause an immune response. And so that leads into the preclinical stage. So this is when researchers assess the immunogenicity of an antigen. So the immunogenicity is the ability to cause an immune response. So does this antigen create an immune response? It's also important during this phase to figure out, is the antigen safe to administer? Will it kill you? So obviously that's important. All of this testing happens in cell culture or in animals, mice or monkeys. It's mostly going to be mice at this stage. Researchers may adapt the antigen during this phase to make it more effective. So if they have an antigen and it doesn't create a really strong immune response, they may change it a little to see if it will then create a strong immune response. So this is also when challenge studies are going to start happening. So this is when they start to learn, is the immune response protective? So is this going to protect people if we use this antigen for a vaccine? So what they would be doing is, like in mice, they would be giving them that antigen with an adjuvant, and then they would wait a little while, and then they would challenge them with the pathogen, just like Edward Jenner did with poor little James Phipps. And do those mice then get the disease? If they get the disease, they know that's not going to be a great antigen to use for a vaccine. So the kind of a key takeaway here is that you can have an antigen that creates a really strong immune response, but that immune response doesn't protect you from the actual pathogen, from the disease, because it's not the right immune response. So this phase typically takes 1 or 2 years. And honestly, most vaccine candidates don't make it past this stage. So this is really tough. They weed out a lot. So again, key takeaway, it's very, very hard to get it right. So once it goes through the preclinical stage, then it goes into clinical development. And this is where you're going to see the phases where they're testing it in humans. So the first one would be, is it safe? And then the very first thing they try to learn is it safe to give to people? Because if it's not safe to give to people, even if it causes a protective immune response, but say it kills it, it creates a protective immune response in half the people that get it, but it kills the other half. That's not a very good vaccine. So if you're paying attention to the news with vaccines against SARS-CoV-2, the virus that causes COVID-19, there's numerous vaccines in clinical trials, and they're all in this stage. So at this point, mostly all we know about them is they create immune response. It may appear to be a protective immune response, but again, this is in mice and monkeys. So mice and monkeys are not people, and they're going to have different immune responses. And so again, this typically takes 1 or 2 years. So the fact that, we're in September and they already have vaccines in clinical trials is pretty phenomenal. It does this way faster than a vaccine has ever been developed, ever. Okay. So getting the right immune response. So it's not easy. This is kind of something I want everybody to understand. The immune system is very complicated. Antigens are very complicated. So it's not easy. So with vaccines, you want to mimic the immune response of naturally occurring infections. But you don't want those adverse effects. So this is kind of the goal of a vaccine, to mimic that infection without actually getting the disease. So some people think, well, why don't we just suffer through the disease? Well, you could suffer through the flu. It still kills people, but how many people want to suffer through Ebola? So this is, again, why we have vaccines, and we want to get that right immune response. So one antigen may be immunogenic enough for long-term immunity, or one antigen may not be immunogenic enough for long-term immunity. So it's finding that right antigen. So you could find an antigen that causes a protective immune response, but it doesn't protect you for very long, like the anthrax vaccine that only lasts a few months. So you would need multiple doses, you'd need boosters later in life, that's something you'd have to keep up with. One antigen, a different antigen, may trigger the right type of immune response to fight the pathogen, or may not. So, sorry, I keep missing where I said may not. So it's that in that exploratory phase, they're going through all the antigens, and they have to identify them, and they have to figure out, are they immunogenic enough? Do they create long-term immunity? Does it create the right type of immunity? So, for example, if it triggers an antibody response but not a T cell response, it might not be effective against the actual disease. And we hear a lot about antibodies in the news, so they kind of get championed as like the most important part of the immune system, but they really aren't always. So a T cell response may be much more important for fighting intracellular pathogens. It always depends on the pathogen. They're always different. But your T cells are really, really important because you can have antibodies, but they're not going to be fighting against intracellular pathogens. Okay, I hope this is making sense. And I just want to go back to mucosal tissues and just talk about this for just a second. So injected vaccines may not trigger strong mucosal immunity. So remember our malt tissues. So we have, these are all the different types of malt. We have nalt, balt, salt, galt. Anything that is mucosa, it's going to have its own lymphoid tissue. Our immune system is kind of segregated in a way. You have the mucosa and you have everything else. So if you get a pathogen that infects the mucosa, so it's infecting some up here in the nasopharynx, it's going to trigger that immunity in malt tissue, or because of malt. So it's going to trigger mucosal immunity. Those antigen-presenting cells that are there in malt are also going to go to the lymph nodes, and they're going to trigger immunity everywhere else. But if you have a vaccine that's just injected in your arm, those immune cells, those antigen-presenting cells, aren't then going to go find malt tissue and train those immune cells that are living in the malt. So you still have some immune response if you get, some mucosal immune response if you get an injection in your arm, but it's not as strong as you typically would get from if your mucosal tissue was triggered itself. I think I worded that really weird. But so just the kind of key thing I want you to understand is that making sure you get mucosal immunity is really important if it's a pathogen that affects mucosal areas like SARS-CoV-2. So this is why with vaccine development against SARS-CoV-2, it's making sure they get it right. This is why it's difficult, and because they have all these extra challenges, not just time, because it is a mucosal pathogen, and our current best ways of creating vaccines are injected vaccines, and they're not going to trigger as strong a mucosal response. And this is also why people think like the flu vaccine doesn't work and they shouldn't get it because it's less effective. That's because it's an injected vaccine. It's hard to get a nasal vaccine just right though. So vaccinology is incredibly complicated because the immune system is incredibly complicated, which I hope y'all kind of started to get an inkling of, if you watched last week's video that was 2 hours long. Okay, so anyway, this is, kind of read over this. If you have any questions about this with mucosal tissue, definitely let me know. Okay, so I'm just going to run through the myths. I know a lot of people probably heard these in the news. I felt like I had to cover them a little bit because we're going to be facing large-scale vaccination campaigns very soon, hopefully, with a COVID vaccine. So the number one myth is that vaccines cause autism. This is not true. This has been thoroughly debunked by multiple studies that have looked at hundreds of thousands of children. Unfortunately, this myth became popular because a scientist, his name is Andrew Wakefield, he published a study in which he used fake data. So he faked his study, but it is still something that people believe. Andrew Wakefield has since lost his medical license, so he is no longer able to practice as a doctor. If you want to read more about this, I can have so much information on this I can send out. But this guy who faked his data has caused a lot of problems because people believed his study that vaccines were linked to autism. All right, another myth, it's not necessary to vaccinate babies. The diseases that vaccines prevent are most deadly to the very young. It is very important to vaccinate babies. And this goes with the next myth, that the vaccine schedule is too aggressive and should be spaced out. The vaccine schedule is determined by decades of medical evidence that show there is an optimal window for when vaccines are most effective. So we're trying to vaccinate babies in that window so they get the best immune response. Having multiple vaccines at the same time, like this current vaccine schedule, it does not hurt the baby's immune system. I've also heard that it's actually bad for the immune system. It's really not. They're giving vaccines at the very best times. And this is determined by, again, decades of evidence by experts. Another one that is really common is the vaccines caused the disease they were meant to prevent. I hear it all the time. I got the flu shot and it gave me the flu. The adjuvants in the flu shot made you feel slightly ill for a few days, even up to a week, but that's not the actual flu disease. You can still get the disease even if you have been vaccinated. That the flu shot is a whole complicated thing because it's not perfect, because the flu changes so much we can't make a 100% effective vaccine against it. But it is still, that vaccine is still going to protect you against the flu. So often people who have gotten the flu after they got a flu shot, it would be less severe. But again, just because you got the flu the same year you got a flu shot doesn't mean the flu shot doesn't work, and it doesn't mean it doesn't protect people who are immunocompromised or like the very old and the very young. Okay. Also, most vaccines are inactivated. So most of the vaccines that are going around, they're being used are inactivated. They're killed virus, killed bacteria. They're recombinant. And the ones that are live attenuated, again, I know that I said that they could revert to wild type. That's only in theory. So it hasn't happened. It's something to be aware of, but it's not necessarily something to be so scared of that you don't get vaccinated. Okay, 5 vaccines contain unsafe toxins. I hope I talked about, I covered this with thimerosal and formaldehyde. These actually make the vaccines safer for you and everybody else. And they're not in high enough doses to hurt us. And they're not going to build up enough. So even like though thimerosal has mercury in it, the amount of mercury it has is so small, and that over the course of your lifetime, it's not enough mercury to hurt you. And you get rid of that ethyl mercury very quickly anyway. And then with formaldehyde, there's more formaldehyde in the fruit you're getting from the grocery store than there are in all the vaccines you'll get in your entire life. Even if you're like me and you get vaccinated for like everything, like rabies and Japanese encephalitis and yellow fever, because I travel and I've worked in labs. Okay, 6, the effectiveness of vaccines has never been proven. It definitely has been. It's been thoroughly proven way back since Edward Jenner first used cowpox to vaccinate against smallpox. So I showed you those graphs at the beginning of this slide where we saw how cases of measles and polio just dropped off. And again, if vaccines weren't that effective, I mean, we would probably still have smallpox and rinderpest. 7, not getting vaccinated only affects you. So we talk about vaccinations and we think about vaccinations often on an individual level. But the thing with vaccines is they're really about populations. So vaccines are there to protect populations, and with that herd immunity. So herd immunity protects the people who can't get vaccinated or immunocompromised. So that a vaccine, like you may get the flu shot and you may get the flu that year and you may not think that it works, so it's not important to get it. But again, vaccines are really, really about herd immunity, and they're about protecting the overall population. There's some terminology. I already talked about these, but I put this at the end in case you forget, because these are very vaccine-specific kind of terms: cold chain, wild type, attenuated, adjuvant, plasmid, and immunogenic. And then that's it for this lecture. If you have any questions, you can feel free to email me. I know that vaccines can kind of be a contentious issue, and I certainly don't mean for this to be contentious. And I can back up everything I have said with data if you would like to see it. If you are curious, I am always happy to do that because I kind of love this stuff. I went to a school where a number of the people, my professors, whole jobs were developing vaccines. So I get really into it. Anyway, that's it for today. I hope y'all enjoyed this, and let me know if you have any questions.